All right. We're doing the eye now. It's really late and I'm tired. I'm sleep deprived, but I still need to do this because it's studying for me. So let's get right into it. So the parts that we're probably going to need to know, the most significant ones are going to be the cornea, the sclera. It's not sclera, it's sclera. The C is silent. The limbus, which honestly, the limbus specifically, it's important, but it's kind of secondary, but you still need to know it because it is important. The ciliary body, the iris, choroid, retina, and lens. So, starting off with the cornea. Oh man, I'm really freaking tired. So, on the cornea, there is 5 layers, and the cornea is pretty much transparent. And it's avascular, which means it's got pretty much no blood flow going through it. It's simply just membranes. So the 5 layers going from outside to inside are going to be the external corneal epithelium. And again, we're always, for these types of layered sections, we're most of the time going to be using the same epi, endo, that type of prefix. So just by the time you see this a couple more times, you're gonna get it. And it's gonna click for you when it gets used in other sections of the human body as well. So after the external corneal epithelium, more towards the inside, we've got Bowman's membrane. Trivia fact, Bowman was the name of the character that Ice Cube played in Black Ops 1. We've got stroma in the middle of the sections of the 5 layers of the cornea. Then we've got Descemet's membrane, which sounds like an Egyptian god. I don't know. The Egyptian, we'll call Decimet the Egyptian god of Histo. And then at the very inside, you've got the simple squamous endothelium. So, that being the cornea, now on the other side, I'm going to, not in the middle, you'll see why in a second, I'm going to write the sclera right here. Move that over a little bit. Now the sclera, its purpose is to provide an attachment site for the ciliary muscle. Now, in the middle, I'm going to put the limbus. Why? Because the limbus actually is where specifically the cornea and the sclera meet. So I'm going to just draw a line between these. Label this outside to inside real quick. And so when the cornea reaches the limbus, the Bowman's membrane portion of it becomes conjunctiva. I don't know why. Could not explain it to you, but now you know that the Bowman's membrane becomes conjunctiva when it reaches the limbus. At least you know that. And the Descemet's membrane, when it reaches the limbus, it becomes a mesh of trabecular processes. Now, moving on to the choroid. So the choroid is actually split up into two layers. You've got the inner choroidocapillary lamina. Choroido, prefix meaning it is within the choroid, and capillary, which are very tiny blood vessels. So inner choroidocapillary lamina. So it's the lamina, kind of like a membrane type tissue that is going to be surrounding the capillaries of the choroid. That's the inner layer, obviously, because it says in the beginning. And then the other layer is going to be the outer branch membrane. Now, if you look at the inner choroidocapillary lamina, it exists. Those capillaries actually exist to provide nutrients to the surrounding area. Now, moving on to the layers of the eye specifically, we are going to have 3 layers, just the eye in general. Not a specific structure, but just talking about the layers of the eye right now is going to make a little bit more sense when we delve into the other parts that we're about to get into. So you've got the outer fibrous layer of the eye, which is comprised of the cornea and the sclera. And you've got the middle vascular layer, which is going to be comprised of the choroid, ciliary body, and iris, and the inner sensory layer, which is going to be comprised of the retina. Let's actually go ahead and take a look at the anatomy of the eye. So this tiny little chamber right here, that is going to be the posterior chamber. So we're going to abbreviate that as P chamber. While this portion is going to be the anterior chamber. And then this disc portion thing, at least the surface of it, not the actual entire structure itself, the surface of this disc portion is going to be the pupil, while its actual structure is going to be the lens. Now, the outermost layer of the eye, the absolute most superficial layer, is going to be your cornea. It's the most commonly damaged portion of the eye. And then this little projection right near the lens, near the bottom of the lens, is going to be the iris. Now, when we were talking about earlier the Bowman's membrane of the cornea turning into conjunctiva, you can kind of see that happen right here, where you see where this part kind of fades into a projection. This projection actually is the conjunctiva. So you can see it actually happen on the diagram. And then this muscle right actually where it's this muscle exactly almost where the cornea turns into the conjunctiva almost, that is going to be where your ciliary muscle is placed. That's really messy. I'm going to maybe use a smaller pen for that muscle part. Cool. Okay, and then now we can get into the sclera, which is going to be the back border portion, and the sclera, where it's where the ciliary muscle attaches. As you can see if you follow it down, the outermost border of the sclera is going to be the episclera. And the back wall, whenever you get an eye exam, this is always when they shine that yellow light into your eye, they're looking back into this portion. This is your retina. And then if you've ever heard the term macular degeneration, that is the degeneration of the tissue right here, where the indent is, right where the arrow is, and that is the macula. And then we've got this space being called the vitreous humor. Now, the chambers actually are a little important. The eye itself is considered to have two chambers. So the anterior chamber houses or is filled with aqueous humor. And the posterior chamber is going to be filled with the vitreous humor. And that is going to be the anatomy of the eye. Now switching backwards, back to the layers of the eye. let's just go ahead and quickly label this. Remember that these two are joined together. It's where they meet. The limbus is where these two meet. Now, let's talk a little bit about the iris. The iris is a very simple but rather important structure. It's not very intuitive or complex, but what it does is pretty important and significant. So, the iris actually blocks light from entering the retina, and it filters it aside from the pupils. So, if you were to say that, blocks light from entering retina aside from pupils. And it accomplishes this via melanocytes. Which produce melanin. To undergo this function. So on top of that, with something else that's pretty important as well, is that the iris actually contains the sphincter, not the booty sphincter, it's the eye sphincter, and dilator muscles to close and open the iris. So if your pupils dilate or constrict, that is the iris constricting or dilating the sphincter or the dilator muscles. So it contains sphincter plus dilator. Muscles to open, close, iris. Cool. And then we've got the ciliary body. I'm kind of going here in order of complexity just so we can kind of build our way up and you know which ones to glance through and which ones to actually focus and somewhat memorize. So the ciliary body is actually going to be made up of 3 different things. So it's going to be made up of the ciliary muscles, I'm going to move these down a little bit, actually. The ciliary process, and the ciliary zonule. So the ciliary muscles, they do something very simple. They just change the shape of the lens. Ciliary process, it produces the aqueous humor that we found in the anterior chamber. So when it's secreted, Actually, a better way to write that. It is secreted posteriorly and it travels anteriorly. And these are just directional terms for its transport. Nothing crazy. And the ciliary zonial, it connects the muscles to the lens. The ciliary muscle specifically. It's kind of implied. You should know that. I'd be a little concerned if you couldn't really ascertain that. Oops. You didn't see that. Nothing to see anyways, honestly. Connects muscle to the lens. And if you actually want to see that up close, if you look in really closely to this diagram, if you see these little projections, like right here, those are the ciliary zonials. That's the stuff that's connecting the muscle to the lens, the ciliary muscle. Now, moving on to the lens specifically, so it helps focus incoming light onto the retina. If you want to get a little technical, specifically the fovea centralis. And the fovea centralis actually specifically is an area that only has cone cells. Cone cells being, they are receptors for color, but we'll get into that in a second. But this area basically increases the visual acuity in the retina. So, based off of your sight, what you're looking at, what you're focusing at, the lens could have different characteristics based on the situation. So, if you were to look at something that's distant, your lens is going to flatten out. Which means the ciliary muscle, the lens, it is relaxing. Whereas if you're looking at something close by or near, the lens rounds. Which means the muscle is no longer relaxing, it is contracting. And, the lens, just structurally speaking, it consists of, the lens fiber, obviously, and the lens cells, the cytoplasm specifically, contains crystalline. It's a crystalline protein. Now, these lens fiber cells, specifically, these are terminally differentiated cells, which means they're stuck in that kind. They cannot differentiate into any other type. They cannot morph. They are the way they are forever. There's no going back. There's no changing. And these cells are the ones that actually bind to the ciliary zonule. So they play a big part in the attachment of the ciliary muscle to the lens. So it's all one big system that's working together. Now, the retina. Fair warning, it's a little complicated, so I'm going to write it out first and kind of explain what the layers are like. And then afterwards, I'm going to go through that with a diagram. I just think that it'll help y'all just learn it better. It helped me learn it better, honestly. So when we talk about the layers, we're not talking about outermost or innermost or most superficial or most profound. In this specific situation, we'd be talking about direction. So one end of the layers is going to be facing towards or heading towards the lens, and the other side is going to be facing towards the choroid and the vitreous humor. So starting from the lens side, so we'll say to lens, we're going to start off, and this is the retina, by the way. So let me just label that. So we're going to start off with the inner limiting layer. And this is the layer that is closest in the direction to the lens. Next is going to be the optic layer. Oh, I don't like that I used a lowercase l on that one. That's gonna bother me. We gotta change that. Optic layer. After that is going to be the ganglionic layer. After that is going to be the inner plexiform layer. After that, these next few are pretty simple. They have very predictable names. Inner nuclear layer. And the histological significance, when we go into it right after this, is going to make a lot more sense based off of the names. So finishing up the inner, now we're going to the outer. So we've reached the other side of this layer, trying to head towards the choroid and the vitreous humor now. So now we're going to the outer plexiform layer. Then outer nuclear layer. And then we also, unlike the lens side, we have a layer that's a little different. It is going to be the outer limiting layer. And then we have the rod and cone, which are both photoreceptors. Rod and cone layer, very important layer. And last but not least, on the choroid and vitreous humor side, the pigment epithelium. So we're just going to go ahead and write to choroid and vitreous humor. Now, let's go ahead and make a little section where we can make notes. Now let's start from the same way we started the list. So on the optic layer specifically, there's a significance. So the axons of neurons converge and form the optic nerve at this layer of the retina. So axons of neurons converge and form optic nerve. Then the inner plexiform layer is our next significant layer. So this layer, it connects the inner nuclear layer, the INL, to ganglion cells. So it's basically linking the INL to the ganglionic layer, more or less. So it's like a transfer layer. And then we have the inner nuclear layer, which contains the nuclei, crazy, of various neurons. The outer plexiform layer houses axons of photoreceptors. While the outer nuclear layer is going to house the cell bodies of the photoreceptors. And obviously, rods and cones being photoreceptors themselves, this layer specifically is going to be the photosensitive portion of photoreceptors. Cool. Now, all of this is being supported by our friendly neighborhood. Wait for it. By our friendly neighborhood Mueller cells. I don't know what they do. I don't know where they're from. To be frank, I don't care. All I care about and know is that they support all of these layers for the retina. Now, getting into the photoreceptors and their layers, honestly, looking at this diagram is going to help a little bit. As you can see the layers here, this top portion going to the lens, this one going down to the choroid and sclera, and the sclera housing the vitreous humor. You can see this portion right here is going to be the limiting layer. You've got the optic layer. You can tell it's a nerve layer because you can see the strands kind of bundling up. Each of these circles, they're cells, they're the ganglion cells, so you can tell that that's going to be the ganglionic layer. Whenever we think of plexiform, we think of striations, we think of fibers, like membranous type of vessels, and you can see that there's a bunch of those here, a bunch of roots, fibers, membrane type ordeal. So that will tell you that that is the inner plexiform layer. Now, inner nuclear layer, right? They're housing the nuclei of various neurons. You can tell they're not entire ganglia cells because they're not as big, but they're still housing the nuclei. Now we've got the outer plexiform layer. Again, it's like the inner, except it's more so towards the choroid and the sclera, and it still has those branches, right? So that's a good way to identify them, their position and what they look like and are made of. Now, we got the outer nuclear layer, which obviously has the cell bodies of the photoreceptors. As you can see, because the cone and rods, these photoreceptors are stemming from the pigment epithelium and they're moving their way up. That would make sense. That would show that the cell bodies of the photoreceptors would be housed in the outer nuclear layer if it's corresponding to the lens side of the retinal layer system. And now you've got the outer limiting layer. Again, it's kind of like the inner limiting layer. It's just a block, easy to identify. And then obviously you've got the rods and the cones, which are your main photoreceptors. And then you've got the pigment epithelium. Remember from the skim, these are simple cuboidal. In this case, they're simple cuboidal pigment epithelium. You can see how they're squared off pretty neatly. So that's that. This diagram will give you a little bit more seconds. It gives you a really good look as to the exact layers and how they work from like a molecular standpoint, a histological standpoint, for understanding the retina and its layers. Now, if we want to delve a little bit into the photoreceptors, which we're going to, that's probably going to be on the quiz. Let's be real. Ooh, that was not the correct tool. Like mentioned previously, you've got two kinds. You've got the rod, you've got the cone. Now, the rod, it tracks light, whereas cone, it tracks color. So, since it tracks light, the rod's primary light sensitive, its primary receptor protein is going to be rhodopsin. And the cone's primary receptor protein is going to be iodopsin. Now, what are these? Well, we'll talk about it. So, rhodopsin is a light sensitive receptor protein. And iodopsin is a light wavelength sensitive receptor protein. Difference is, when rhodopsin gets activated, it's light sensitive. It detects the intensity of the light that is hitting its receptors. Whereas iodopsin, we see color in wavelengths. So, the higher the wavelength, the more deeper the color. So, for example, like violet would have an extremely high wavelength, whereas red would have an extremely low one. So the wavelength differentiation is how we can differentiate the different colors: red, blue, green, yellow, whatever. So iodopsin being the cone's main, light sensitive receptor protein, it's going to detect color. So therefore, its main indicator is going to be light wavelength. So that's the difference. And I'm going to flip over to the diagrams real quick, but I think that's about it for the eye. I guess I lied in the chat. Ears took a lot longer, I think. But I don't know. I think the eye is a little bit more complex, and I would spend a bit more time on it. But thank you for my recording. Thanks for listening.